Hello everyone, welcome. So this is class 12, microbes in human welfare. So we do find microbes everywhere. So they are present at the sites where no other life forms are even present. Okay, so that as they are even present at a very high temperature at 100 degrees Celsius deep inside the soil, and microbes are diverse, so they can be protozoa, bacteria, fungi, they can be microscopic animals and plant viruses, varioids. So there are different types of microbes that is there. So here we have bacteria, which can be rod shaped, it can be spherical, it can be flagellated, whereas we have viruses, which may be bacteriophages, adenovirus, which causes respiratory infections and all those. And we have these colonies of bacteria and fungal colonies as well, which we see over here. Now microbes, they we are talking about the microbes which is important for the human welfare. So microbes in the household product, if we are making curd, so there is the lactic acid bacteria, lactobacillus, which is there, so which grows in the milk and convert it into curd. During the growth, the LAB produces acids that coagulate and partially digest the milk protein. So a small amount of curd, when added to the fresh milk as inoculum or starter, contain millions of lab, which at suitable temperature they will multiply and converting the milk into curd, which also improves the nutritional quality by increasing the vitamin B12. The dough which is used for making foods used such as dosa, idli, they are also having fermented by bacteria, which is due to the production of carbon dioxide, they are getting puffed. Okay, so similarly the dough which is used for making bread is fermented using baker's yeast, that is Saccharomyces cerevisiae. A number of traditional drinks or foods are also formed by fermentation of microbes. So today, a traditional drink of some parts of southern India is made by fermented sap from the palms. Microbes are also used to ferment fish, soya bean, bamboo, etc. Cheese is one of the oldest food items in which microbes were used. So there are different varieties of cheese which is known based on the texture, color, taste and all those things. Next, for example, if we talk about Swiss cheese, they are due to the production of large amount of carbon dioxide by bacterium named as Propionibacterium shermani. Okay, then we have the Roquefort cheese, which are ripened by some fungi. So likewise we get the flavors. Then we talk about the microbes in the industrial products. So in the industrial products we have fermenters. Fermenters, the production of industrial scale, requires growing microbes in very large vessels, which is known as fermenters. You have the fermenters over here that you see. Then we have the fermented beverages. What are the different fermented beverages we have? We have wine, beer, whiskey, brandy or rum. These are one of the beverages we have. For this purpose, Saccharomyces cerevisiae, which is used for bread making, which is known as brewer's yeast. Okay, they are used. They are used for malting. So, this fermenting the malted cereals, fruit juices, and all those things. Depending on the type of raw material used for fermentation and the type of processing, different types of alcohol drinks are obtained. Then we have antibiotics. Antibiotics are produced by microbes, which are largely regarded as one of the most significant discoveries of the 20th century. Okay, we are talking about antibiotics. So antibiotics are produced by microbes and are regarded as one of the most significant discoveries of the 20th century and have greatly contributed towards the welfare of the human society. Anti means against and bio means life. So together when we say antibiotic, it means against life, life of the disease causing microorganisms, whereas with the reference to human beings, they are pro life and not against life. They are towards the life. So antibiotics and chemical substances which are produced by some microbes can kill or retard the growth of other microbes. So there are a lot of things. So Alexander Fleming, when he was working with staphylococci bacteria, he once noticed that a mold growing in one of the unwashed dishes, unwashed culture plate, uh, around the streptococci, around which streptococci could not grow. So he found that, by chance, he found that it was penicillin. He named the matter as penicillin and the mold as Penicillium notatum. Okay, so that is how the antibiotic came into example. Full potential as an effective antibiotic was established much later by Ernest Chain and Howard Foley. The antibiotic was extensively used to treat American soldiers during the World War II. That is when antibiotic came into play. Chain and Foley, they were awarded the Nobel Prize in 1945 for this discovery. Okay, so after penicillin, other antibiotics were also purified from other microbes. Okay, so antibiotics, which were against plague, whooping cough, diphtheria, all those things were also manufactured. Okay, next, chemicals, enzymes and other bioactive molecules. So there are various chemicals. So microbes, they are used for commercial purpose and industrial production of certain chemicals like organic acids, alcohol, enzymes and etc. So examples, you can say producers like Aspergillus nigeria, which is a fungus which is used for producing citric acid, Acetobacter aceti for producing acetic acid, then Clostridium butylicum for butyric acid, and Lactobacillus for lactic acid. Yeast, which is Saccharomyces cerevisiae, is used for commercial production of ethanol. Then microbes are also used in production of enzymes. Lipase are used in detergent formulations and also helpful in removing oily stains from the laundry. So here, if you would have noticed, the bottled fruit juices bought from the market are clearer as compared to that made in the home. That is because of they are being clarified by pectinases. Streptokinase, which is produced by the bacterium Streptococcus and modified by genetic engineering, is used as clot busters for removing clots from the blood. Another bioactive molecule which we have as cyclosporin. Cyclosporin A, that is used as an immunosuppressor agent in organ transplant patients. In the organ transplant patients, is produced by the fungus Trichoderma polysporum. Stains produced by yeast Monaskis purpurus has been commercialized as blood cholesterol lowering agents. So there are different enzymes, there are different bioactive molecules or chemicals that are being produced. Next, we come to the microbes in the sewage treatment. So within the sewage treatment, there is the sewage treatment plant, the STPs. Okay, so here there are different types of treatment that takes place in the sewage treatment plant, like primary treatment, secondary treatment, so on and so forth. So primary treatment, where there is the physical removal of particles large or small from the sewage through filtration and sedimentation. These can be removed in stages. Initially, floating debris is removed by sequential filtration. Then the grit, they are removed by sedimentation, and all solids that settle from the primary cell sludge and supernatant forms the effluent, and this will undergo the secondary treatment or the biological treatment. So here the primary effluent is passed through large aeration, where it is constantly agitated mechanically and the air is pumped into it. These allows vigorous growth of useful aerobic microbes into flocks. While growing, these microbes consume the majority of organic matter in the effluent. So this significantly reduces